So Dr. Moore did her PhD at North Carolina State University. She's done a couple postdocs at North Carolina State, University of Montana, University of Denver. And another cool thing about Emily is that we have both sort of crossed paths in like weird ways at several times. Both have had connections at Montana, and then we both appreciate the African Great Lakes, although our appreciation has manifested in different ways. And so on that note, I will pass it on to Emily to talk about between species and sexes, the genetic basis of adaptive traits in African cichlid fishes.
Well, thank you, and thanks to Jess and Renee for inviting me. I have been here for just a little over a year now at UNL over in Mantor Hall, so if you want to come visit me. And I do have an aspect of my research that I'm not talking about today, but if you're at all interested in reproductive biology and the placenta, you can also talk to me about that, so at a different time. So when I think about sort of one of the fundamental driving questions of my research program is this idea of how do we get so much biodiversity? And my guess is that some of you are also interested in this general idea. And one of the groups that I've been working with is African cichlid fishes. And this is just a smattering of the species. There are, thousands of species of cichlids, many of which are in the African Great Lakes. And these are just a few that I have personally worked with in my career. And when we look at this diversity, some of it is a little bit surprising. So for instance, these three are different individuals of the same species of the same population. They're just have different sex chromosomes. And then, for instance, these two, even though they sort of maybe look similar on first glance, they've got similar coloration. They're actually from different genera. They have different jaw morphologies. They have different life history strategies. And so there's a lot of ways in which biological variation is represented within this large group of fishes. And we're at a really cool time right now where we are able to generate a lot of data. And this data is allowing us to find the underlying genetic basis of many different kinds of traits. But a lot of this work is being done in classical models. So like mice, zebrafish, fruit flies. We have a lot of tools in these sorts of organisms. And I think what I really want to be able to do is to take these kinds of tools where we can really get at mechanism and bring them to more non-models like cichlids so that we can not just catalog the genetic variation in cichlids, but actually tie how these changes to the genome are actually generating adaptive traits so we can get a better picture for how species are actually functionally able to adapt to their environment. And so one of the things that I've been really excited about doing is to look at tissue architecture and gene expression to be able to get better picture of what mechanisms are tying these to genetic variation to phenotypic variation. And so sort of a lot of the questions that I ask in the lab start with natural history. So thinking about water quality, thinking about what are the actual habitats these animals are living in. We do genetic strategies to find the underlying genetic basis, but then go in and look at the brains, look at specific brain regions, look at different parts of different organs, and then tie that to differences in cell type and what genes are doing within those cells. Okay, so if we go back to the picture of our fish, these are fish from the African Great Lakes, which Jess alluded to. They're actually the second largest lake group in the world, rivaled only by the American Great Lakes. So think about lakes with their own weather systems, right? Like these are enormous bodies of water that have a lot of ecological opportunity for the fish that live there. And my work focuses majority on fish from Lake Malawi. in large part because they're sort of the Goldilocks lake for geneticists, where in Lake Victoria, which is this top lake, we have a lot of young species that are, about 500,000, half a million years old. In Lake Tanganyika, which is this middle lake, you have a lot of older species lineages where you have, there may be 15, 20 million years diverged, and you only have a couple examples of each lineage. And so it's harder to do some of the comparative work. But in Lake Malawi, you're in this like perfect spot where you have a lot of trait differences between species, but then the genetic relatedness allows you to do a lot of between species comparisons and in the lab crosses. They've also, in Lake Malawi, adapted to a wide variety of habitats. And so for the purposes of this talk, I'm going to simplify, but know that this is indeed a simplification, and I recognize this is a simplification. But you can broadly think about it on a sort of benthic to limnetic axis, where I'm going to call fish that are species that are adapted to the rocky shore, rockfish or rocky fish. We have species that are adapted to sort of intermediate zones at the rock-sand interface, and then other species that are more adapted to open water type environments. And when we think about the ways in which these animals have adapted to their environments, like these are truly very starkly different environments. So here's a picture of a lek of breeding open water male cichlids that have all built a bower or like a little sand castle that they're defending to attract mates. And so they build species-specific patterns of sand bower and then do species-specific patterns of dances on top to attract the right female. And this is clearly very different from this sort of heavily inhabited, many species interacting, highly dense, rocky landscape. So there's a lot of differences in the ways in which you have both interest-specific and interspecific interactions. And this has led to selection for differences in male color. So we have a lot of differences in species differences in nuptial coloration and mating strategy. There's also huge differences in foraging strategy and resource use. So these open water fish are using these sensory pits and they're sensing invertebrates under the sand, whereas a lot of fish that live at the rocky shore are either algae grazers or they go into the water column and get zooplankton from the water column. And these dietary differences are reflected by craniofacial morphology. So these are cartoons that I drew, but I meant to sort of really illustrate some of the large differences in craniofacial morphology. So this top one has a big old schnoz that's used to actually physically lever algae off of rocks. And so they have really huge changes to both the buccal jaws, they're like teeth like our teeth, and then they have pharyngeal jaws, which are the teeth like moray eels or like the alien in the alien movies have that come out of their throats. And they have a huge adaptations to both of those jaws. But one of the places where I feel like I've been trying to make inroads is this idea that there's lots of different, also differences in environmental novelty. So whether you're going to see a lot of dynamism in the environment, as is seen in this rocky shore, versus a big open water, like very open to predator habitat, as you would see in a sand habitat. And so what I've wanted to test is not behavioral responses in a mating sense, which has been historically looked at in the group, but actually like, can we detect the behaviors that are allowing for like adaptive changes to differences in niche? And so I've been working on this, I guess, for the last 7-ish years. And I found a few ways that we've been testing it in the lab. So this is an example of one where we just place a novel object in the tank and see what the fish does. So this fish really likes the novel object. It says shell, it's moving it around. And what we can do is trace the position of the fish over time. So each of these dots is one spot where the fish was in a frame of a video, and this is a snapshot of 5 minutes of what this fish does. So this fish has a self-imposed radius where it is not investigating the novel object. Whereas this fish goes up and spends a lot of time investigating the object. I think it may have actually moved it around. It may be that fish that I just showed you. And so we can capture through this sort of computer vision strategy clear differences in adaptive or exploratory behavior. And so my first question is, do we only see these facets of differences between species? Or are these also facets that are underlying sex differences within the groups? And so I'm using this really cool system where we know we have diversity across sexes. And to tell you this story, I'm going to need to start off by talking a little bit about the genetics of sex determination and sex chromosomes. So to understand how sex chromosomes generate diversity in traits, we first need to think about what sex chromosomes are present and what are they doing. So when I'm talking about sex chromosomes, I'm talking about a genetic switch that is inherited and can help determine the fate of the gonad and secondary sex traits. So for us, for humans, and for most mammals, we have something called an XY system. And I'm using a Y in a very similar way, where it's going from fathers to sons. So it's malely transmitted. It's sort of a male switch. We can also have genetic sex systems that we call ZW systems, and they're very similar, but instead of the switch going from fathers to sons, the switch goes from mothers to daughters, and we call that the W. So I'm going to use the same terminology where we have either a female switch or a male switch, but unlike mammals or birds, our cichlids don't have degenerated or very small sex chromosomes. They actually have spots on regular autosomes that begin to function like sex switches. So instead of having a very small W, there is a whole chromosome that just has a spot on it that sometimes functions as a W or a female determiner. The other thing to note is that these chromosomes are present in the same population, which means that given the right pairing of individuals, you can have an offspring that inherits both sex chromosomes. So instead of two genetic sexes like we have, where we have either XX or XY, there are actually 4 genetic sexes with this pairing. And so I've done some work where we made this pairing in the lab to ask, how does the presence of 4 genetic sexes alter secondary sex traits, and what does that mean for the evolution of these sex chromosomes? Well, the first question is probably that you might have is, are fish that get 2 sex chromosomes one sex or the other? What I'm showing you here is if you inherit a W, if a fish inherits a W chromosome, they make eggs. And then if they inherit a Y chromosome, they make sperm. So these are sort of our standard male-female gonad histology. So females who do not inherit a sex switch at all make eggs. And an individual that inherits both chromosomes also makes eggs. So what that tells us is that we have 3 genetic ways to make eggs and 1 genetic way to make sperm. However, just even by looking at the fish, we can see that there is something more complicated going on, and that is that sex is linked with color. So for fish that have a W chromosome, they have this orange blotch, like blotchy or mottled color. And this first is actually the first way we found as scientists that this polygenic sex system was happening. And it's because, so male nuptial coloration, as I alluded to earlier, is really important for these fish. It's how they signal, I'm the right individual to mate with. It's also how they show health. It's like kind of an honest signal. If your color is nice and bright, you're probably great to mate with. And this orange blotch makes males really ugly. Females don't like them. However, this blotch was proposed to be a way to help female crypsis. And so you have this something called sexually antagonistic selection, where blotching is really bad for males, but really good for females because it helps them hide. And here's some actual photographs of actual blotching fish. And what we found is that larger blotches tend to be associated with spots where there's bigger pebbles, and smaller blotches tend to be associated with spots in habitats where there are smaller pebbles. So we actually have this sort of range in levels of blotching that background match within females, supporting that it's crypsis. So here I've added in our smaller blotch female over here in the diagram. You may say, oh, well, sure, they're blotchy, but they're bright orange. Well, it turns out that red wavelengths of light penetrate water the least well. And so for us, when they're out of the they look orange, but actually under their water, they're basically invisible. So we have the system where we have light blotching on more sandy background at the interface and more heavy blotching where it's more rocky. And we have that segregating for both orange background fish and white background fish across the lake. And one of the things that we wanted to know was, is this something that has just happened once, or do we repeatedly have small blotches evolving? And so if we look at our population, majority of their genetic signal splits them by location. So all of the white blotching fish grouped together and all of the orange blotching fish grouped together. But then when we look just this spot in the genome, they group by how blotched they are. So both the white and the orange blotch, light blotch grouped together, and the bigger blotches grouped together. So what that suggests to us is that selection on color for background matching can actually move sex chromosomes around the lake. So you have selection pushing different alleles of sex chromosomes through different populations based on the traits that aren't about sexual or aren't new sexual antagonism. And so it's not about the sex differences in the trait that are moving the sex chromosomes around. It's the ability for those alleles on the sex chromosomes to resolve differences in adaptive landscape, or like adaptation to background. So the whole reason I went into this story is then we can think then, like, are there other traits that are reshaped by the presence of sex chromosomes that allow for adaptation? And what does that say about how sex chromosomes are going to move around populations, and how those sex chromosomes can remodel organism level traits to allow rapid adaptation to environment? So things that I've looked at so far, I looked at sex differences in behavior. So when we take a fish and we place them just in a home territory, we can ask how long do they, from the beginning of the assay, how long do they stay inside their little cave, and when do they start to come out? So what I'm showing you here is that males spend less time in their cave at the beginning of an assay, and females spend more time hiding. And then as the assay progresses, everybody starts to come out and spend time outside. So the females are hiding a little bit more, they're a little less exploratory. When we add a novel object to the tank, sort of like simulating an environmental disturbance, females with a Y start to leave their territory earlier than their females that don't have a Y, and they do it at the same rate as their brothers. So by having females with a Y chromosome, we've suddenly made a female that has more resilience to environmental disturbance. So then you can imagine, if we fix that Y in the population, we've now made a population that is more resilient to environmental disturbance. We also see that the combinations of sex chromosomes remodel head morphology, which, as I said earlier, is very important in dietary adaptation. And so we can quantify our shapes using geometric morphometrics and show that, so males are more different than all of the females, but then females with a W grouped together versus females without a W. And then the next axis of variation is actually separating out females with a Y from females without a Y. So we're able to actually expand the range of base shape variation by adding and mixing and matching which sex chromosomes are present in an individual. Okay, so then given this, why are some traits sexually dimorphic in some species but not in others? Well, when we have these kinds of polygenic systems, or you might call it a multigenic system, it's just historically called polygenic, it allows us to remix traits as sort of a binned or modular way with different outcomes if you fix different combinations of sex chromosomes. So if we start off with this set of 4 possible combinations, but then we lose the novel W, we end up with colors that are less different from each other. We end up with behaviors that are more different from each other, versus if we say fix the Y in everybody and then just use the W as the sex switch, we then have females that always look much different than their males. And we have behaviors that are more similar and more robust to change. So we can actually alter whether traits are sexually different in these populations by fixing or one set of sex chromosomes or the other. And then when we look at the traits as a whole, that can tell us a little bit about mechanism. So for quite a few traits that I looked at, we actually don't see any difference based on chromosome. So this whole suite of traits seems to be mostly influenced by hormones or gonadal state. But then we have sets of traits that are W-linked, where we can imagine that depending on whether we fixed one set of chromosomes or the other, we would swap whether that species is fixed for one trait or the other. Vice versa, with Y-linked, we have some situations where now the species does not have sexual dimorphism, or it does. And then the coolest thing is that when we talk about these epistatic traits, so ones where it matters which combination of the sex chromosomes you get, we can actually switch the valence of the trait, which means that the trait that was once predominantly in females becomes predominantly males in the other population, just based on which sets of sex chromosomes you keep in the population. So it allows for really rapid remodeling of whole traits in organisms and allows for switching back and forth about whether a species has sexual dimorphism or doesn't. So these new combinations of sex chromosomes can both generate and maintain diversity across the group. We know that other species have fixed these chromosomes in the ways that I'm suggesting. Like, it's not just sort of theoretical, we know this has happened. So I'm really excited in the lab to, as my research keeps going here at UNL, to keep looking and identifying for these species that naturally fix these traits, do we see the same evolutionary outcomes as we might expect, given this model of like fixing or maintaining sex differences. And we're currently collecting brains and reproductive tissues from this cross so that we can look at gene expression to see how are the sex chromosomes interacting with hormones to remodel what sex differences in gene networks are doing. So how are we actually generating sexual dimorphism and traits in these crosses? Okay, so sex chromosomes are really cool, and they're really important for generating this within species diversity that seems to be important for allowing cichlids to diversify across the lake. I alluded to earlier how important or cool it is that we can also identify species-specific behaviors. And I've had the ability to do a deep dive on quite a few of these species and highlight here a few of the species that I'll tell you a little bit more about. But first, we needed to figure out which are the behaviors that actually track with habitat use. And so I worked with a lab at Georgia Tech, and we sampled all of the species represented here, and we measured exploratory behaviors in several ways for everybody. And this is just indicating whether the species is specialist, a SAN specialist, or an interface specialist. And this is a rough phylogenetic tree. And while we looked at a bunch of different traits, the thing I'm going to show you today is this open field assay, where what we do is we take the fish, we place it in the middle of a kitchen supply big white container, and we track where it goes, where fish that spend more time in the center would be in sort of mouse or classical behavioral assay prevalence, like more exploratory, and then fish that are on the edges and hug to the edges a little bit less exploratory. And we actually can replicate this within our group. So we have some fish that tend to swim more quickly and stick around the edges, and then some fish that swim more slowly and tend to explore the middle of the arena a little bit more. And so there's this sort of inverse correlation. You are either, if you spend more time on the edges, you're spending less time in the center, and vice versa. And so when we look at this time in the corner, so that's the inverse of time in the center, we can show that these open water species tend to spend a little less time in the corner, and then rocky shore species tend to spend a little bit more time in the corner. And so we can capture these microhabitat differences in behavior in the lab with this like high throughput computer vision based kitchen supply store white bin assay. And so we can use these lab results as a proxy for habitat associated behaviors. So the next thing to do is to figure out what are the actual species differences that are underlying exploratory differences. And so to that, I use classical quantitative genetics, which is one of the superpowers of cichlids. We can actually take a rock species, in this case it's called Metriaclima and Bengii, and a sand species, which is Alanacar koningsi, and we can crop them in the lab. And so we measure their behaviors. So here our Metriaclima and Bengii is our low exploration fish, our Alanacar is our high exploration fish. We make F1s, where they have 50% of their genome come from a rock species, 50% come from a sand species. And then we use the magic of recombination to remix the genomes in our F2. And then we measure behaviors in every single one of these F2. We then go through and we scan the genome to ask, where in the genomes do fish that have the rock allele have the more rock-like low exploration behavior, and where do they have the sand allele and the more sand-like high exploration behaviors? And what this looks like statistically is you group everybody based on what genotype they have at the locus, and if that spot has a signal, you'll have a positive association there, and if there's no signal, you have no association. So that's the sort of statistics we're looking at. And when we take swimming speed, so how fast they're going, we can look at that statistical signal that's represented here on the y-axis is LOD score, or logarithm of odds. This is just saying how likely is it that that is associated with our behavior. And then we plot that for every chromosome in the genome, and we can find that swimming behavior has a complex genetic basis. So for the 331 fish that we measured, we can detect 4 spots in the genome that seem to be contributing to how fast they're swimming. And 2 of them are really associated, and 2 of them are more slightly associated. And chromosome 5 has the strongest signal. So from this point on, we're focusing on chromosome 5. So if we zoom in on chromosome 5, this is what the signal looks like. We've got a bunch of markers here along the chromosome, and this blue region is the part of the chromosome that's associated with our behavior. So swimming speed maps here. Also, center position maps here. So we have the same chromosome that seems to be influencing both swimming speed and whether or not they're going in the center or the edges. But you may notice it is a very large part of the chromosome, and there are many genes in this region. And so it doesn't really get us to the genetic basis quite yet. Here we can go to the other superpower of cichlids, which is that they breed intermittently in the wild. And so we can go and survey population genetics from the wild and bin those species or populations by the trait we're interested in, and ask, where do our rock and our sand populations from the wild differ from each other? So what we're looking for is this metric called FST. That's a comparison between our populations, where we might, for a lot of the genome, see that there's no difference between our rock and sand group. There's no allelic variation here at all. Maybe a variation, but it might not have been by rock versus sand. And what we're looking for is spots in the genome where all or most of the rock species have one version and all or most of the sand species have another version. And so we can take our map and we can plot on, overlay onto it, these population level differences to zone in on the spots where we both have a signal in the lab and a signal from the wild. And that narrows it down a lot because of historical recombination. And so when I zoom in on this right here, that is both high FST and under our peak, we wanted to go back and look at a broader set to really ask, like, sure, this seems like it's associated with habitat, but what if that's just a spurious signal because we didn't have that many samples to begin with? So we looked at 39 genera and 82 species to ask, are they segregating the variation, or are they fixed for the rock or the sand allele here? And what we find is that all of the species that are fixed for our rock allele, 60% of them are rock specialists. Whereas if we look for the ones that are fixed for our sand allele, only 15% of those are rock specialists. So we have a huge enrichment for sand specialists when they are fixed for the sand allele. We also see that there are species that are segregating the variation. They have both the rock copy and the sand copy at this locus, and 55% of them are interface specialists. They're adapted to be at the rock-sand interface. And within this group, it's representing 3 different genera. So repeatedly, we see evolution for our selection for maintaining genetic variation at this locus for fish that live in between the rock and the sand. And so fortunately, we had a few of these in the lab, and in the lab, they were segregating the variation in the lab. So instead of making just a cross between species where we have those genetic differences, we could actually look in a population or where the majority of the difference between our individuals as we were binning them was this particular variant. So we can take our heterozygous individuals for this species is Metriaclima aurora, and then look at full siblings that are the same age, grew up in the same environment, and that just differ based on this SNP about whether they have the rock allele or the sand allele at this variant. And so I did this for both Metriaclima and Trophiops that were segregating the variation. I redid the kitchen supply assay, and as you might expect, I'm telling you about this, and so we replicated the difference here. We also coolly could do things like this, where we made a rock sand microcosm to make it a little bit more naturalistic than a kitchen supply container, and ask, do full siblings choose to spend more time on the rock side if they have the rock allele, and vice versa? And they do. So for siblings that have the rock allele here, they have a higher preference. They spend more time on the rock side of the arena than if they have the sand allele. So we can replicate this microhabitat choice in the lab in these interface species. Okay, so I promised you expression networks, and I'm only going to get to a little bit of it because the future of my lab is going to be looking at this sort of expression network stuff. So we found this. I've hopefully convinced you it's associated with behavior, and that behavior is relevant to microhabitat differences. What does this spot do? Well, it's a single nucleotide polymorphism, and it's upstream of 3 neural cell adhesion molecule genes, or NCAMs. And so it's not in a gene, it's a non-coding element. And the genes, all 3 of these genes are really important for axon guidance and patterning. So they've got these cool little immunoglobulin domains on the outside, and they help axons know where to go as the brain is being patterned. However, beyond that, we don't actually understand that well how these particular genes remodel or repattern axon guidance. And so I think that that's exciting, and like that's a thing that we can start to learn a little bit more about using cichlids. The other thing is this region, it may be non-coding, but it is a conserved non-coding element. And it's conserved all the way to zebrafish. Zebrafish are 200 million years diverged from cichlids, so it is striking to have a regulatory element that's conserved for that long. So that tells us that that probably has a function. The other thing we can see here is that the rock allele is the same allele that's fixed or conserved to zebrafish. So then we can say something about which one is the new allele. It's the sand allele. Our sand allele is our derived allele, and we can start to make some hypotheses about this region. So if we take the sequence and we look in a database, it turns out that it comes up as the top 8 hits are for neural transcription factor binding, which this all makes sense. But it can help us make this hypothesis that this G allele here is actually in some ways breaking transcription factor binding or reducing the efficiency of transcription factor binding. And so by having a G allele here, we've reduced the efficiency of our transcription factors for this site. And so we would expect to see this in how the genes are expressed in the brain. So we have our ancestral allele, we expect to see some level of gene expression. And then with our derived sand allele, we expect to see a lower level of expression because we've broken the transcription factor binding. However, there are lots of reasons that gene expression in the brain can be different between individuals. It can be, slight differences in experience or hormonal cycling. Like there are a lot of reasons why gene expression in the brain is really liable. And so what we can do is look in individuals that have copies of the SNP. So we're heterozygous individuals, and we can ask for an individual that has both copies and the exact same brain, is one copy more highly expressed than the other? And if we were looking at just the genome, we would expect it to be 50-50 because they have one copy of each. And what we find is that for CHL1, the rock copy is expressed on average twice as high as the sand copy in the same individual. So this supports our idea that we've broken transcription factor binding at this site, and we have higher expression of our rock allele than our sand allele. Other cool thing about this is it turns out that a CHL1 mutant, this gene, has almost no phenotype in mouse, except that it makes the mouse spend more time in the center of an open field. And because it's conserved all the way to zebrafish, we decided to do some functional tests using doing CRISPR in zebrafish because it's a little bit more easy to do than it is in cichlids. So we did functional confirmation by making 2 different CRISPR lines. I did this with a collaborator, where we knocked out the gene itself, and then we made a line where we knocked out the regulatory region. So then we could ask, can we replicate or make zebrafish behave a little bit more like cichlids with these knockouts, replicating that reduction in expression? And what we find is that when we knock out CHL1 entirely, we replicate the center phenotype that we also see in mice, where they spend more time in the center of an open field. And when we knock out the conserved non-coding element, we change the speed phenotype. So we can actually, by modifying these different elements of this locus, replicate both the speed and the center position behaviors in zebrafish. So we have a single locus that seems to be modulating a complex behavior using these sort of multiple mechanisms. And it seems to be a case that conserved mechanisms are underlying these habitat choice and use behaviors. So when we are learning about them, we can perhaps be learning things that could be more broadly applicable about how the brain is working and about how habitat choice might be happening. And fundamentally, this niche partitioning behavior, like are you spending more time in the rock, more time in the sand, these are behaviors that may help eventually maintain biodiversity because they're actually physically shifting where animals are spending time, and you're reducing the probability of overlap and potential interbreeding. So with my last couple of minutes, I just would love to tell you a little bit about some of the projects that we have ongoing in the lab that I've been doing with undergraduates, and we have incoming wonderful graduate students who are going to be working on some of this. We have other things that we're measuring about our fish. One of them is body shape and tail shape. So we know that life history strategy, like tunas versus gobies, have huge differences in how they swim. But it's sometimes really hard to compare the genomes of a tuna and a goby and get it just the differences in body shape or body plan or tail shape. And when we look at zebrafish are really great for understanding mechanism, but they only have one type of tail unless you make mutants. So we can take advantage of the fact that cichlids have different tail shapes and tie that to both the genetics and the performance. So that's what we're doing. We're looking, in this cross, we've been able to show that there are differences in tail shape and that those differences in tail shape are genetic. So our F1s are sort of intermediate between forked and fan-tailed. And here is a picture of some of the F2 showing the range of diversity in tail shape from our very forked sandfish to our very flat-tailed rockfish. And we can describe this with principal components and show here's the range of variation of our mapping cross. And we're finding genetic sites associated with tails. So we're able to understand the genetic basis of this morphological trait. But then the coolest part is then we have behavioral data for all of these individuals. So we're just starting now to look to see whether tail shape is associated with behavioral differences. And this is just a subset of the animals because we've only started reanalyzing the behavioral videos in the way that helps us see this. But we're using this postural tracking with a program called T-Rex, where you can see here that you can actually see when the fish is beating their tail. So I want to be able to try and capture the moments where the fish is beating their tail to see whether tail shape versus body shape is actually contributing to these differences in swimming performance. And Hendy is going to be working on that. And then the other thing that we have going on that may be interesting to you is we're also looking at the genetics of otolith size. So otoliths are inner ear bones in fish, and they help maintain balance and posture in addition to being able to be used to age them, which is pretty cool. And so these are really important for spatial perception. And it turns out they are very enlarged in this, as you can see in this bottom fish, in our open water species. And so we've looked at these otoliths for a variety of different species. This is a very similar plot to the one I showed you previously, where we have our open water fish on one side and our rockfish on the other, where open water fish have much larger otoliths. Our sandfish have much larger otoliths than our rocky shore fish. And we have not one, but actually two crosses where we've been able to map this trait. So we're identifying regions that are contributing to this sort of otolith and balance type trait. And it turns out there's a very, very slight signal between how fast they're swimming and how big their otoliths are. So the idea is here that we can look at all of these different components that influence behavior from all of these neural, morphological, balance type ways to integrate to understand how you shape a behavior. So we're understanding these complex behaviors from both an like morphological and neural perspectives. And hopefully it's giving us a little bit of an insight into the origins of this biodiversity and mechanisms that underlie how traits are generated. And so with that, I'd like to thank my lab, the people who've been working on this project so far, my collaborators at NC State and Salish Sea Research Center, and you for coming to this talk. And thank you so much.
That was an awesome presentation. And I know Ethan sitting next to me, he said he's really into cichlids. So it's just really neat to see. And I'm not familiar with genetics really, but I'm just curious if you've ever visited these Great Lakes in Africa or have plans to in the future.
Yeah, that's a great question. I was supposed to go in grad school. I had a whole landsect experiment where I was going to go from the sand all the way through the rock and sample along it and collect behaviors along the way and look at allele frequencies for different populations. And then we had a total fish room collapse, and we had to shift all of the resources away from going to the lake to ordering more fish in. And so I've actually never gone. I do have a collaborator at the University of Wyoming, Katie Wagner, who studies Tanganyikan cichlids, and she does a lot of field work. And so she and I have been talking about ways that we could work together so I could go out with her and her crew. And so I would love to in the future, definitely. Like, be amazing to see them in the wild.
And this might not be a question you know the answer to. So obviously African cichlids are also very, very popular in the aquarium trade. And do you know, because where are you getting your cichlids shipped in from the wild, or are you using aquarium bred cichlids? That's question one.
Yeah, that's a great question. Definitely wild derived.
Okay.
So most of the work that I showed you is from popular lab lines that were collected by my PhD advisor. And so we know specifically exactly where those fish are from. But then we also work with, there's like one fish importer who's really reliable. His name is Dave. And so we get our fish from Dave, and Dave can say these are, what generation they are from wild caught, and they are, have their specific location. And so we're somewhat limited based on the locations that he can get fish from. So we aren't sampling from the entire lake, but most of the pet trade cichlids, that unless you go to like a cichlid hobbyist convention and somebody who's like keeping lines in their basement or something, like when you go to PetSmart, those are all mutt fish. They are perfectly wonderful, lovely pet fish, and that there's no assessment about the pureness of quality of them based on their pureness, but they are not fish that I could do science with.
Right. Well, that actually leads to my second question. So way back when I was in my undergrad, the master's student that I was working with her project was looking at sailfin mollies, but she was looking at wild caught versus aquarium trade mollies and behavioral differences in mate choice copying. Is there anything that you could potentially do to compare wild caught cichlids versus changes to behavior in aquarium bred cichlids, and the potential genomic background behind that?
Yeah, I mean, there's all kinds of reasons that I think that happens. There's, you select for the fish that do the best in the aquariums when you bring them in, and the fish that are maybe the most choosy maters or the most aggressive that are causing issues and that you can't keep with other fish, they get purged from your breeding stock. And so you're automatically imposing a selection pressure even from the very beginning for the fish that you can keep in the lab. And then you have the ones who are successful at breeding. So you might have, other types of traits that influence behavior, but that are reproductive that you're selecting for. And then there's the aspect of you're in a different environment, right? Like the lab is definitely very different from the lake. And so just even if you have the same individual fish before selection, it might behave differently in the lake than you would in the lab. And I think that that's just a thing that we have to accept is the caveat, right? Like if what we want to do is understanding the genetic basis of the behavior, then it is better for us to accept that we will have purged a little bit of variation bringing them into the lab, but that we have the ability to control the environment a little bit more finely so that we can reduce what we call environmental variance. And I have thought a little bit about this, like how much do we want to, like like what are we sacrificing in our ability to say things about the wild and what's going on in the wild? And I think I've come to the conclusion that, you know, as a geneticist, I've like claimed that the thing I want to find is the genetic basis of things. And so, yeah, that's it. It's a sort of a tough philosophical question. It's a good question. I like it.
Thank you.
Yeah.
Hi, Emily. So you started out saying that Malawi is sort of this Goldilocks between Tanganyika and Victoria. I've been to both Tanganyika and Victoria. But the thing that I'm thinking about with Tanganyika is if the fish are more ancient there, does that change your expectations of how much of the like squishiness in genetics that you see there? Like, are the species so well defined that?
Yes, and we're talking about, you know, having maybe, so there's something like maybe 18 or 20 different species that are tropheus. So tropheus in Tanganyika is the haplochromine lineage. So that's the same lineage that's in Malawi. And so even for that lineage, we see a lot fewer representatives of it. And then you have certain lineages where there is only a single extant species from that lineage. So like Scypha tilapia frontosa, there is only one of those. That species is the only representative of its lineage. And so I think of it as like, if I were guessing, you know, Tanganyika probably had this expansion with a lot of interbreeding, a lot of squishiness, and then the lineages that were the most successful have now survived whatever pruning happened. And maybe that's not going to happen to Malawi, right? Like it's hard to know, it's hard to predict what's going to happen. But it means that we have a lot less, individual species have sort of narrowed down and then are maintaining the variation that they're maintaining.
And then because I'm a person that thinks about the water and the environment in the water, I can't help think that this is such like a visually based system, and that like the African Great Lakes, not Victoria so much as Tanganyika and Malawi, are so well known for their water quality and that like clear water clarity.
Well, and Victoria has been being pretty well studied for the past, I don't know, 20 years-ish, because there's an increase in eutrophication. And so a lot of the signaling is visual, as evidenced by having mating displays, building these big bowers, having these bright colors. And in Victoria, because there's an increase in turbidity and decrease in water quality, it's allowing for species collapse, because you have these nascent lineages that would have been successfully assortatively mating that no longer assortatively mate because they can't see who's who. And so there is this idea that potentially we could see some sort of, probably not in Tanganyika, but potentially in Malawi with some of the species, we could see a reduction in sort of lineage diversity, although we would maybe see maintenance of genetic diversity if we had a decrease in water quality. Though they are a little picky about who they mate with. So even in the lab, sometimes it's hard to get them to mate, and we have to strip eggs and then squeeze milk onto them. So we have to do like a poor man's in vitro fertilization. Or sometimes we have to do a love the one you're with strategy, but it has to be like, he has to be the only male on Earth, you know, essentially. Like he's the only one in the tank. Sometimes we'll have to like put a male that she likes in the tank next to her so she can see him. So at least she's like thinking maybe a little bit about mating. Right? Like there's a lot of finagling we have to do. And so the fact that it's sometimes so hard to get our hybrid crosses to go makes me think that like Malawi might be a little bit resistant to reductions in water quality in terms of those sorts of visual signals, but it's hard to say.
Anyone else? Might just check.
Okay, so we've got, you refer to different species. By what concept are these fish separate species? So I would say the ecological species concept. So they hybridize intermittently. You can hybridize them in the lab. Like they live in different areas, they eat different things, they have different morphologies. And so I think that, like, I personally think of species as a continuum anyway. And so I'm not particularly tied to any one species concept. So I don't mean to be flippant about that question, but I think they're act as pretty good functional species insofar as that they're able to maintain their genetic variation separate from each other most of the time. And then I have, okay, what number and effect of genes underlie the adaptive shape differences and rapid diversification of cichlids in African lake species or even in the laboratory setting? And how does genetic variation for these traits evolve over time? Okay, well, every trait has a different genetic basis, and even, well, it depends on which particular pair you cross, you know, whether or not that allele is there generating diversity often not just depends on, you know, that allele, it also depends on which other alleles are present with it. They're all interacting, and so it's hard to have a hard and fast, like, this number of genes or this number of alleles affects the trait. So for the particular cross that I showed you with behavior, with the number of individuals that we measured, we were able to detect 4. But there are probably more, a lot of small effect loci we couldn't tell. And I'm sure if we made a different cross, and we could see this with the otolith cross, like sometimes you see the same spot over different crosses, and sometimes you can have the power to detect a new spot. And so most of these are polygenic or multigenic. They have like multiple interacting spots across the genome. So that gives us a chance to really see how genes work together to make these traits. And then we can see for a lot of these traits, the genetic variation is maintained more broadly across the lake. So this is one of the cool features of cichlids, is that you have the same adaptive allele selected again and again. So cichlids aren't reinventing the wheel within the lake most of the time. You know, if you see, you know, particular jaw shape in one species, it often has very similar genetics as that same jaw shape in another species. And it's that repeated intermixing, remixing over time that allows them to sort of mix and match. And then selection can act upon that suite of traits that are best for that particular little region in the lake. And so you have this ability to sort of churn a little bit to get the options to have the best suite of traits. Well, best, I mean, it's hard to say if it's the best, but you're constantly churning just a little bit so that selection can keep acting, but maintaining enough difference from each other, a lot of interbreeding, so that you have time for those alleles to go to high frequency without new alleles coming in. So it's a really cool evolutionary system, and I think for that reason it makes cichlids really great for studying these types of traits. So those are great questions. Thank you.